So we are on to our next lesson for this chapter. And again, at the end of this lesson, hopefully, you guys will be able to functionally define cellular respiration, identify the reactants and products of cellular respiration, differentiate the major chemical events of cellular respiration, and summarize in the form of illustration or diagram the process of cellular respiration. Okay, so this time we will be discussing about how eukaryotic cells produce most of its ATP in the mitochondria. So glycolysis always occurs in the cytosol, as what we have discussed during our synchronous session, but the location of the other two pathways in aerobic respiration depends on the cell type. In bacteria and archaea, the enzymes of the Krebs cycle are in the cytoplasm, and the electron transport proteins are embedded in the cell membrane. The eukaryotic cells of protists, plants, fungi, and animals, however, contain the mitochondria, which is a specialized organelle that house the other reactions of the cellular respiration. A mitochondrion is bounded by two membranes, which are the outer and the inner membrane, which is highly folded. Cristae are folds in the inner membrane. The intermembrane compartment is the area between the two membranes, and the mitochondrial matrix is the fluid enclosed within the inner membrane. So all of those parts you can actually see here in this diagram. So you have here the inner membrane, here is the cristae, you have here the matrix, the intermembrane space, and we also have the outer membrane. So, in eukaryotic cell, the two pyruvate molecules produced in glycolysis cross both of the mitochondrial membranes and move into the matrix. Here, enzymes cleave pyruvate and carry out the Krebs cycle. Then, FADH2 and NADH from glycolysis and the Krebs cycle move to the inner mitochondrial membrane, which is studded with many copies of the electron transport proteins and ATP synthase. The inner membrane's cristae provide tremendous surface area on which the reactions of the electron transport chain occur. Electron transport chains in ATP synthase also occur in the thylakoid membranes of chloroplast, which generate ATP in the light reaction of photosynthesis, which we have discussed in the previous chapter. So if you forgot, this is how it looks like, or this is how the ATC happens in the chloroplast. Now, similar enzymes operate in respiring bacteria and archaea, making ATP synthase one of the most highly conserved proteins over evolutionary time. Now, mitochondria and chloroplast share another similarity. Both types of organelles contain DNA and ribosomes. Mitochondrial DNA encodes ATP synthase and most of the proteins of the electron transport chain. So, not surprisingly, a person with abnormal versions of these genes may be very ill or even die. The worst mitochondrial diseases affect the muscular and nervous systems, and muscles and nerve cells are especially energy hungry. Each one may contain as many as 10,000 mitochondria. When their mitochondria fail, these cells cannot carry out their function. So at this point, we will try to account for all the ATPs that are produced in the whole cellular respiration. So the question is, how many ATPs can one glucose molecule yield? Now here is an image which will show you the amount or the number of ATP, NADH, and FADH2 that are produced during the entire cellular respiration, and also the accompanying table for summary. So let's discuss it one by one. Now, to estimate the yield of ATP produced from every glucose molecule that enters aerobic cellular respiration, we can add the maximum number of ATPs generated directly or indirectly in glycolysis, the transition step, the Krebs cycle, and the electron transport chain, as you can see here in the picture. So you have here the glycolysis, the transition step, the Krebs cycle, and the electron transport chain. Now, Glycolysis yields 2 ATPs, as you can see here, as does Krebs cycle. Okay, so that 2 ATPs produced in the Krebs cycle, take note, there is 1 ATP each from 2/3 of the cycle, so that gives us 2. In addition, each glucose yields 2 NADH molecules here in glycolysis, and 2 more from acetyl-CoA production, or in the transition step. Now, 2/3 of the Krebs cycle yield an additional 6 NADH and 2 FADH2. In theory, the ATP yield from the electron transport is, as you can see here is the conversion. For every 1 NADH, that is equivalent to 3 ATP, and every 1 FADH2 is equivalent to 2 ATP. So look here. So you will see that electrons from the 10 NADHs from glycolysis and the transition step and the Krebs cycle therefore yield up to 30 ATPs. So you have here 2 plus 2 plus 6, so that's 10. So remember, every 1 NADH is equivalent to 3 ATP. So 10 times 3 would give us 30. So in ETC, we will have 30 ATPs. Now, it does not stop there. We still have to add the electrons from the 2 FADH molecules, which yield 4 more. So you have here the 2 FADH, and according to the conversion table, every 2 FADH is equivalent to 2 ATP, so that gives us 4. So 30 plus 4 gives us a total of 34 ATP, but we have to still account for the other ATPs produced in Krebs cycle and also in glycolysis, so that gives us a total of 38 ATPs. However, NADH from glycolysis must be shuttled into the mitochondrion, usually at the cost of 1 ATP for each NADH. This reduces the net theoretical production of ATPs to 36 ATP. So, you might notice I used the word theoretical, right? So, in reality, some protons leak across the inner mitochondrial membrane on their own, and the cells spend some energy to move pyruvate and ADP into the matrix. Now, these expenses lower the actual ATP yield to about 30 per glucose, and the number of calories stored in 30 ATPs is about 32% of the total calories stored in the glucose bonds. The rest of the potential energy in glucose is lost to the environment as heat. This may seem wasteful, but for biological processes, it is reasonably efficient. To put this energy yield into perspective, an automobile uses only about 20% to 25% of the energy contained in gasoline's chemical bonds. The rest is released as heat. So you can look at it like that.